gotten to meet a lot of really nice and kind people. And then the science convenings have been really helpful for discussing and communicating about my research and just kind of getting the creativity flowing. In science, you do a background search in what's already known, and based off of what's known, you try to generate the next big question, and ultimately these things lead to discoveries that actually have an impact on human health. I've been studying neural circuits involved in cancer, cachexia, and anorexia. So that condition in cancer patients, many cancer patients develop loss of appetite and muscle wasting that results from inflammation from the cancer. And that's actually one of the primary causes of morbidity in cancer patients is the systemic wasting of organ tissue. We don't actually know the exact causes and mechanisms. Where we actually started was we started in a brain area that we knew was important for controlling appetite and food intake. And it was really an area that's important for transmitting those signals also into a general affective emotional state. So when you eat something, there's usually a feeling that's associated with it. If you're too full or if you have food poisoning, you feel ill. And we hypothesized that this brain area, because of all the different types of sensory inputs that communicate that something is bad is happening, we hypothesized that it would be critical for cancer, anorexia, and malaise associated with it. So the neural circuit that we study is actually really important for transmitting a visceral malaise. And that visceral malaise, when paired with the taste, actually starts to form aversive memories. And that's one reason why cancer patients, especially those going through chemotherapy, develop an aversion to some of their favorite foods. And so that's the problem with cancer, is that a lot of patients lose that innate drive to consume food and to maintain their body weight. So these neural circuits that are involved and important for transmitting this aversive signal that causes loss of appetite, they're in part modulated by descending control from my higher brain areas. So there are psychological treatments that could actually help with treating the symptom. At the same time, there are signals that are intrinsic and circulating throughout the body that counteract that innate drive to consume food and maintain body weight. And so that requires not only identifying the neural pathway, but figuring out how to actually inhibit those aversive signals that lead to body weight loss. The brain area that we discovered that was critically important for loss of appetite and feelings of illness, it's actually very deep within the brain, so it's actually not very easily accessible pharmacologically. And so a lot of what my future research is going to be on when I start my research lab is to figure out how the neural circuits eventually impinge onto this brain area that we discovered, and to try to figure out easily accessible pharmacological pathways that we can inhibit this brain area. My graduate school mentor, he played a huge role in helping me find the right mentorship and the right fellowship. So his name is Robert Ritter. And then I joined Richard Palmetter's lab for my postdoctoral fellowship, where his lab primarily focuses on neural circuits using a mouse research model, and we use genetics to parse apart all the different neural pathways. And while I was in Seattle at the University of Washington with Dr. Palmetter, our lab also collaborated with Dr. Michael Schwartz, who's in the Department of Medicine. And it was through that collaboration that we honed in on this particular area of research. Our lab, and due to the support from Hope Funds, we actually were the first to discover a discrete group of neurons in the brain that is critically important for cancer, anorexia, and cachexia. Since our publication, there have been follow-up studies from other researchers as well as pharmaceutical companies that have identified upstream pathways that lead to activation of the neurons that we implicated. It requires a lot of passion and dedication, but it's also very rewarding. So our brains are made up of neurons that transmit sensory signals throughout the brain, and that leads to a computation of how to interpret the sensory signal and what to do. That means that different neurons in different brain areas perform different functions depending on the type of information they received and what sort of motor pathways are connected to. That means that cells genetically express unique proteins because of their specialized function. And we use genetic methods to hone in on those particular pathways. So they're sort of like they're like labels that we use. So cells express different things, and we take advantage of that using genetic tools to manipulate them, to visualize them, and then to activate or inhibit neurons. I actually started my undergraduate at Eastern Washington University in Cheney, Washington, as a Running Start student during high school. From there, I transferred to Washington State University to finish my undergraduate school. During undergraduate school, I was really inspired by physiology and biology, and my initial passion was actually to become a doctor to help people that have disease. As part of that experience, I got to do research in Dr. Robert Ritter's lab, where he studied gastrointestinal mechanisms of satiation and neural pathways that relay to the brain signals that tell you to stop eating during a meal. I really enjoyed that research, so I continued on during graduate school and got my Ph.D. there in neuroscience. From there, I went to Dr. Richard Palmer's lab at the University of Washington, where I was interested in using advanced genetic tools to parse apart different brain neural pathways involved in behavior. Because of my background in neural circuits controlling appetite, and because of Richard's interest in a pathway that was involved in suppressing appetite, we focused on a population of neurons that potently suppress appetite. And these neurons had been implicated in responses to visceral malaise, so signals that cause food poisoning would turn on these neurons and cause anorexia and loss of appetite. And so that was when Dr. Michael Schwartz, my co-mentor, hypothesized that possibly these same neurons would be involved in cancer and loss of appetite and body weight loss associated with cancer. And so that's sort of the origin of the project that I was working on. It's both narrow and wide, so you have to take inspiration from different areas of research to come up with a very specific hypothesis. So one example of this was the population of neurons that we were studying. These neurons had been implicated in responding to visceral malaise signals during food poisoning, and then I later implicated these neurons as being involved in gut signals that tell you to stop eating. And then there was a postdoc in Dr. Palmer's lab that discovered that these same neurons that are involved in control of food intake were also turned on by foot shock. So somatic pain relayed through a totally different sensory pathway. And that was part of the reason why we hypothesized that these neurons would be critically important for cancer, anorexia, and malaise, was that there was a confluence of very different aversive sensory pathways that turn on these neurons. And since we didn't know what was the exact cause of anorexia and malaise in cancer, we thought that we could just go into these population neurons that already were known to integrate diverse signals. And so my area of research, even though it initially started in the neural circuits involved in the control of food intake, it essentially expanded to include other sensory modalities that also turn on these neurons. So Hope Funds has allowed me to continue my research past graduate school. This is during my postdoctoral fellowship, so this is a critical period. It's analogous to medical doctors who, after finishing their MD, they go on to residency to do their final training before they can become certified doctors. And so Hope Funds has allowed me to take that next step. And through the support from Hope Funds and the research that bore out of it, I've essentially carved out a new area of research that's really critical for treating cancer patients. And I'm going to be establishing an independent lab based off of that. So one thing that's really important about Hope Funds for cancer research is not only the financial support that allows me to actually conduct the research, but the foundation is important for collecting scientists and for bringing them together at various events, either through social events or science convenings. And those are critical interactions where we are surrounded by people that are very encouraging and have a certain youthful optimism to their approach. And in science, we go through struggles like any other profession that is worth working on. And that has really helped to support that mentality. And so that's something that I'll be carrying forward when I start my own research lab. As young scientists come into my lab, they go through ups and downs, and I think it's really important to have that sort of support with young people as they start off in their career.